Hi, dear students. In this lecture, we will see what is the tissue culture media and what is synthetic media and what is the difference between natural and synthetic media. What are the growth regulators used here and other components of tissue culture media. I think you are already aware there are two types of medias. One is ready-made media and another one is natural like conventional method. We have to prepare separately growth regulators, macronutrients, micronutrients, and also gelling agent. Everything we should separately add. And ready-made mixture is the one which can be directly used. They will recommend always like 34.7 grams in 1 liter of water. So sometimes it will be having it will be having only the media like major and like minor nutrients. Sometimes it will be also having growth regulators. There are different medias like in synthetic media itself there are different medias are available, which is mainly for shoot initiation as well as for root initiation. Like you have already practiced all these things in practically, like we have purchased a lot of like tissue culture media preparation things from high media. I think you have seen practically also. In this lecture you will be learning what is media. Media is the it is a like anchorage, or it is a substance which is which helps in growth of the tissue culture plantlets. Like it is similar to soil, but it is artificially prepared. Normally, whatever the media composition or whatever the media we are preparing, that is a very basic thing for the successful production of the tissue culture plantlets. If you are preparing the media pakka, then you can able to achieve the success and you can able to easily multiply the plantlets. But you have to be very cautious about the concentration of macronutrients, micronutrients, and how you are preparing the growth regulators, and how you are handling the like other substances like iron substances should not be added directly. Some chelating effect will be there, so all these things you should consider before preparation of the media. A proper prepared media will define the whole tissue culture success, like success of the whole tissue culture process. So we'll see here like media, we can see both organic as well as inorganic based on the content, whatever the contents present in that, whatever the nutrients present in this particular media, which helps in the cell division, cell elongation, and also the proper growth and development and production of shoots, roots, other things. And also whatever the media which is the content, chemical content as well as the physical, it should be like liquid or solid or semi-solid condition for performing various types of tissue culture techniques. In practical number 8, that is media composition of common tissue culture media, you have learned whatever the contents in tissue culture media and how much quantity of macro micronutrients should be added and other things you have already studied. So here we will see so what is synthetic medium and what is natural medium. So normally the synthetic medium or synthetic media is defined as the media which contains all the chemicals except yeast or animal or plant tissue. No yeast or animal product or plant tissue is present in the synthetic media. It contains all the like elements in a inorganic form. So next is natural media. Some of the natural medias like vegetable extract can be used, fruit juices or plant extract can be used as a media. So this is called as a natural media. Even coconut water can also be used as a natural medium. It contains cytokinin or kinetin, which is also one major growth regulator which helps in successful production of the tissue culture plantlets. Kinetin or cytokinin also we can use. So we will see which are the different types of mediums or media. So normally solidified media. So it will be the, as the name itself indicates, it will be in a solid form. And what is the advantage of using this particular solid medium? Like we can easily suppose explants, if uh, we can easily place the explant and we can easily recover. We can, if there is any problem, we can easily recover. And here there is no like excess air, no need to provide the excess aeration. And shoot growth, it is normal and it is in a orderly manner we can expect. And we can keep it for long time. And also we can use some small containers. Even in the small containers we can use this particular solid medium and we can easily handle. And the problem here is solid media, the rate of multiplication will be less here. Suppose from one explant we can get around some 4 to 5 the multiplications. But here that is a one disadvantage. And whatever the explant which we are using, it is not having much contact with that medium. The surface contact is very less, I can say. So this is the one disadvantage here in case of solidified medium. Next one is liquid medium. Liquid medium normally callus formation or for callus culture they will use. So here the callus easily break and it will establish very fast. So multiplication rate will be more and surface area of the explant will be more and contact, like whatever the disadvantage you have seen in the solidified media, where it will not have much contact with the medium. So it can be avoided using liquid medium. Like toxic metabolites, easily we can sterilize this also. Like whatever the toxicity is there, that can be easily sterilized. So that is a one advantage. Next is we can also see some disadvantages here. Recovery is difficult. Suppose when the explant, if there is any, suppose any damage, that recovery is difficult. And growth also we cannot expect. Like solid medium means it will hold, the anchorage will be good, but here it is not the case. And if we are placing any seeds in this liquid media, it will not germinate. It is well suited for callus culture. And the protocoms and the plantlets become brown. Like whatever the protocoms or plantlets we are using, it will become brown. And it will show the symptom of vitrification symptom, because of the lack of aeration or because of the lack of movement. And the plant cannot hold the media cannot hold the plant for long time. So these are the some of the drawbacks here. Solid media, normally we will use agar as a solidifying agent. In case of liquid medium, we can use like here normally they will not suggest any gelling agents. Here only like gelling, because of the absence of gelling agent only, the medium will be in liquid state. And we can also see some protocom like spherical structures here, like especially in case of orchids, they have beautifully depicted. You can see here in the liquid medium, like test and other things are present, and it is the final product. Like this the from starting to the whole plant development stage in different stages in our orchids. So this is like you have seen with like solid as well as liquid medium, and how it will be having lot of effect on the success of tissue culture process. Normally, Murashige and Skoog is the one universally or widely used medium. So it is available both as like ready-made as well as conventional method we can use. So nowadays, all the tissue culture ventures are the units wherever the commercial practice is followed. They are all using uh, ready-made medium only. Ready-made medium, it is easy, so we can avoid the problems while weighing, and also we can avoid the inaccuracy and other things we can avoid easy. It is easy compared to the normal or conventional method of preparation. So this lecture may helpful for you to know like which type of medias are available, and what are the medias will be suitable for different kinds of thing. Normally solid media we can use it for almost all the plantlets. Suppose if we are concentrating on protoplast culture or callus culture, we can use liquid medium as well. So liquid medium will also helps in like those kind of things, like especially for those kind of protocols it is very useful. So this is about media preparation, and also like components of the media, and which are the different types of mediums available, and so what is solidified media, what are the advantages, disadvantages, and liquidified medium or liquid medium, what are the advantages and disadvantages. So this is about the entire like medium. Already you have learned this practical in eighth practical. So this is kind of recap for you people. Okay, thank you. If you have any doubts, you can ask me.